Here, in a continuation of the previous short lecture, we are continuing with the terminology again, and in that we have discussed about the gene and gene in a particular previous class or previous lecture. In this, we are going to talk about allele, and for allele, understanding allele, we have to understand locus first. So, what is locus? Locus is a position of a gene or chromosome. So, the gene, wherever the gene is located, that is a locus, a position of that particular gene. So, suppose these are the homologous chromosome. Okay, you can see, and in homologous chromosome, this is the particular position of a particular gene. Okay, and this is a particular position of another gene. So, this position is known as gene locus. Okay, so this is the same locus. This and this are the same position at same position. These two genes are at the same position. Means we can say this gene 1 and this gene 2 are present at same locus. So if we talk about a locus, it is the position of gene on a homologous chromosome that we have discussed. See, some of the examples are given here, and this example shows that alleles of gene A are A, capital A and small a, they are present at the same locus. There is the same locus. C at the same locus, B at the same locus. So this capital B is the allele. This capital C and small c are a particular allele pair. So what is allele? That is the question. So allelic pair on homologous chromosome at their respective loci is located in this diagram. So we have to understand what is allele or what is allele. Allele is also known as allelomorph. Now, two Mendelian factors or genes which occupy the same locus on the homologous chromosome that control expression of a particular character, then it is called as what? A alleles. Means two genes are there. Suppose capital T and small t, or capital A and small a, that are present at the same locus on the homologous chromosome, and they code for the same character. Then that are known as alleles. The question is, example is given here that genes of purple color flower and white color flower present on the same locus on the homologous chromosome. Means they have two alleles for each character or each gene. So this is, you can see here, this is allele of a purple color and this is allele for a white color. These are the homologous chromosome and this is the same locus, okay, for flower color. So for flower color, if we represent purple color by a capital P and white color by one small p, means capital P and small p are the alleles of each other. These are the two alleles which are coding for the flower color, and these two are the alleles of a gene P, okay, or a gene which is coding for the flower color. So because of this, we call alternative forms of allele genes are known as alleles. Alternative forms of genes are also known as alleles. So write up one definition. Alternative forms of gene which code for same character are known as allele. Now, what are the units of inheritance? Gene is the unit of inheritance that we have discussed. Now, after this, the initial some discovery was started, but Mendel proposed that something was being fast, and by the introduction of Mendel, we actually start a genetics. So Mendel was a scientist who proposed that something was being passed down unchanged from parent to offspring through the gametes. Means parents produce a gametes, that gamete fuse and produce a zygote, and the zygote form a next offspring. So through gametes, it is passing to the next generation from parents to the offspring in successive generation. And he called these things as a factor. What Mendel called factor. Nowadays we call them as a gene. And we have discussed gene are the unit of inheritance. This gene contain information that is required to express particular trait in an organism. If you want to express any trait, for each trait there is a particular gene. What are alleles? That also we have discussed. Genes which code for a pair of parent contrasting trait is called alleles. They are slightly different from the same gene. If we use alphabetical symbol for each gene, then the capital letter is used to trait the expressed trait expressed at the F1 or a hybrid stage and the small alphabet of the other trait. Whichever get expressed in the F1 trait according to the law of dominance, then it is a dominant trait, and the particular trait which is not expressed in the F1, that is recessive trait. So for example, in case of the character of height, capital T is used for the tall trait and the small t is for the dwarf trait. Capital T and small t are the alleles of each other. Means these two alleles calls for the character height. So after this discussion, we can conclude that hence in plant the possible pair of alleles for height would be capital T, small t, pair of allele, capital T, possibility, capital T, capital T, capital T, small t, small t, small t. Okay, so this is the thing. Now, when this pair of allele come in, when alleles come in pair, and when same type of allele are forming a pair, then it is known as homozygous condition. And this is also representing homozygous condition, but this is representing heterozygous condition. So heterozygous condition show the pair of alleles. So homozygous gene pair carries two genes for a trait. For example, a polled cow might carry a gene pair capital P, or horned cow must carry the gene that is small p small p. For a cow to have a horns, she must carry two recessive genes. Small p is a recessive gene and capital P is a dominant gene. You can see these are the some of the characters: detached earlobe. All these are genetically correct, continued. See, this is the thing: extra finger, detached earlobe, attached earlobe. See, so here I want to make you understand two terms, that is a genotype and phenotype. Genotype is the what a particular gene genetic constitution which is responsible for the phenotype, and they can be homozygous recessive, heterozygous, or heterozygous recessive. So if we want to express what is genotype, genotype is a genetic makeup that code for a trait, that is a genotype. And if we talk about a phenotype, phenotype is physical appearance of trait. So what physically is seen, either it is a tall or a dwarf, that is particularly shown here. And see, here the example of detached earlobe and attached earlobe is given. If there is a attached earlobe, then it is a recessive condition. Both genes are in a small condition. So this is genotype. But what is genotype? Attached earlobe. For detached earlobe, it can be either capital E or capital E small e. So heterozygous show the detached earlobe. Next, we are going to talk about this stem condition: homozygous, heterozygous, and hemizygous condition. So, if we want to describe this and we want to write a definition of this, then homozygous means homozygous is condition is presence of similar alleles of a character in pair in diploid organism. See, diploid organism means presence of two set of chromosomes. Okay, so two set of chromosome means one is maternal set, one is paternal sets. If we draw the chromosome like this, total 8 chromosome are there. But suppose these are maternal chromosome and the remaining are the paternal chromosome, means which these are received from the father and this are received from the mother, and they are forming a pair. So this maternal set and paternal set, two set are there. So it is known as diploid. But if there is only a single, they are not in pair. So it is a single set and it is known as haploid, and gametes are always haploid. So in diploid, where the two set of chromosome are present in this, suppose the particular gene is present and gene are of same allele in pair. Suppose here capital T and here capital T. Okay, then it is a homozygous condition. So we can write example capital T and capital small t. But another is the heterozygous condition. In heterozygous, presence of dissimilar alleles of a character. In heterozygous, there is a it is a pair of dissimilar alleles in diploid organism. Okay, so it is a 2n condition, and if we write for example, then it is a capital T small t condition. Okay, that is heterozygous condition. Another term remaining, that is a homozygous condition. So if we write hemizygous condition, then what is this that we have to discuss? Now, hemizygous means presence of single allele. There is no requirement of pair here. Here, presence of single allele of a character in diploid cell or organism. So in a diploid cell or organism, if there is a presence of single allele, then it is known as hemizygous condition. Example, we can say the X and Y chromosome. In if we take X and Y chromosome, then Y chromosome is somewhat smaller. Okay, so this is X, this is Y. On X and Y, there is some homologous region, and there is some non-homologous region. So on homologous region there is a pair of allele, but in non-homologous region, the alleles which are present here, or the genes which are present here, and the genes which are present here are not coding for the same character; they are coding for the different character. So hemizygous is the single allele of a character, presence of a single allele of character in diploid organism, and that single allele is present on that single allele is present on non homologous region of a X and Y chromosome. And if we talk about the particular gene like this which has a single allele, that is TDF factor. This gene TDF factor or TDF gene, that is a testing determining factor, it is present on Y chromosome only. So it is linked to the Y chromosome only. So we call it gene as a hemizygous gene. Okay. So this is the thing about the particular, you can say, hemizygous condition. So after discussing this homo, hetero, and hemizygous condition, now we are going to understand some more terminology. Okay. Then we understand one more term, that is phenocopy. Okay. So next term is phenocopy. Now this phenocopy, if we talk, then it says that there are two genes which produce normally different phenotype. Okay. If they produce a similar expression due to external factor, then they are known as phenocopies. Okay. So what is phenocopies? When two different genotypes or genes which normally produced different phenotype, which normally produce different phenotype, if they produce similar expression, due to external factor, then they are known as phenocopy. Okay. Means phenotypically they are copying the gene. Actually they are two different genes which are present on the non-homologous chromosome. Okay. But they have a similar expression. Because of this, they are phenotypically similar. Phenotypically they are copying to the each other, so they are known as phenocopy. Now next we have to write a dominant trait and recessive trait. Dominant trait, if we talk, dominant trait is a trait which express in both homo and heterozygous condition. If they express both in homo and heterozygous condition, then it is a dominant trait. If we take example, capital T or capital T small t, this both having a phenotype that is a tall, means the either in homozygous condition, that is capital T or heterozygous capital T small t, the phenotype is going to be tall only, means tall is the dominant trait. The another trait which we are going to talk, that is a recessive trait. And if we talk about recessive trait, this recessive trait specifically expressed only in homozygous condition means if there is a small t, then it can be only dwarf. In capital T small t, dwarf will not be produced. So this is a recessive trait. Okay. Now, after this, if we talk about some more terminology, then some more terminology is the offspring. So we remember, dear students, offspring are the progenies of a parent produced by sexual reproduction. Okay. Then one more terminology that we are going to discuss, that is genome. Dear student, genome means genome means it is a sum total of all the genes, all the genes present on haploid number of chromosome. So sum total of all the genes, it is just total sum of all the genes which are present in a haploid number of chromosome. Suppose we have a 46 chromosome in human being in a one cell. The 23 chromosome combinedly form a one set, and that 23 chromosome will represent a haploid number of chromosomes. So all over the gene which are present on the haploid number of chromosome, that form a one set or one gene, one genome. So haploid cell having a one genome, diploid cell having a two genome, triploid cell having a three genome. Like this we can say. Okay. So let's move further. Apart from this, the one more terminology which we are going to discuss here, that is a gene pool. If we talk about gene pool, it is a sum total of all the genes, sum total of all the genes of all the organism of a population. So if we take whatever the all genes which are found in a population in each individual, that combinedly form a gene pool. Then the next terminology which we are going to discuss here after gene pool, that is a inbreeding. Inbreeding, if we talk, it is a breeding between between closely related closely related members of a species. Means if one species, individuals of one species are there which are closely related, belong to the same species, if they are breeding, then it is known as inbreeding. And inbreeding is a one type of selfing, means we are breeding of a plant with the same type of plant, with the same type of another plant, then it is known as selfing or inbreeding. So this selfing, which is known as inbreeding, it increases homozygosity. It increases homozygosity in progenies. Okay, so that is inbreeding. Another is the outbreeding. If we talk about outbreeding, outbreeding is a breeding between between distantly related distantly related members of a species. Okay, so that is outbreeding. And outbreeding increases heterozygosity in progeny. It increases heterozygosity in progeny. So inbreeding, in the continuous selfing, outbreeding, it is the breeding between the differently related organisms. After this, we are going to talk about some more terminology, and in that, we talk about hybrid vigor. Hybrid vigor, which is also known as heterosis or shell. Now, what is this thing? First, understand, dear students, you know that if we talk about our father, or it is your father or my father, they are comparatively superior than their fathers, and we are comparatively superior than our parents in some aspects, not in all way, in some aspect. So whatever the superiority of offspring occur over the parents, it is known as heterosis or hybrid vigor. So it is defined as it is a superiority of a hybrid or offspring over their parent is known as hybrid vigor. And this variation in the offspring compared to parent, this superiority of offspring over the parent is maintained by outbreeding. Now, so when we perform outbreeding, the superiority of offspring is going to be increased over the parent because new variation are introduced. But vigor decreases by inbreeding. So inbreeding decreases hybrid vigor. that is known as inbreeding depression. Means we lost the superior character due to the inbreeding, because inbreeding is not going to induce a variation. So continuous selfing will decreases the superiority of offspring or a hybrid over their parent, or a variation, it loses a variation, so it is known as inbreeding depression. Then after this, we'll talk about some more terminology like eugenics. So next term is eugenics. So eugenics, if we talk, it is deal with the improvement of human being, and it is improvement of human being by use of principles of inheritance. So by using the principle of inheritance or Mendelian principles, if we carry out this, improvement by the principles of inheritance, then it is known as eugenics. Then the second terminology is euthenics. And euthenics tells that it is improvement of human being by improving their environment. So if the human being get improved due to change or improvement of their environment, then it is known as euthenics. And euphenics is also a one study related to the genetics, that euphenics is the improvement of human by use of medical technology, or genetical engineering. So this improvement of human by the use of medical technology or genetic engineering, then it is known as euphenics. Okay, so these are the three branches which is of genetics which are dealing with this human improvement aspect. Now, apart from this, next we are going to talk about another few terms, and we are going to talk about the branches of genetics. And genetics are of two types. Genetics are of two types. One is forward genetics and one is backward genetics. And another one is the backward genetics. Backward genetics is also known as reverse genetics. And forward genetics, if we talk, then in forward genetics, what it is a study from phenotype to genotype. Means first we study the phenotype and then we talk about the responsible genes or responsible genotype for that phenotype. So whatever the Mendel did in his experiment, where he also noted some morphological activity and then they found phenotypes. So first study of phenotype occur and then a genotype is discovered, then it is known as forward genetic. So example is the Mendelian and Morgan genetics. But backward genetics, if we talk, it is a study from genotype to phenotype. Means first we identify the gene, we locate it, then we talk about its morphological function or the trait for which it is coding. So first we talk about the genes, then we talk about the phenotype. So in this example, if we talk, then it is a genomics or we can say modern genetic engineering is there where it is a type of backward genetics. Okay. After this studying of forward and backward genetics, the next thing which we are going to discuss, that is penetrance. Okay. So dear students, I hope you all are getting these points. Now, when we talk about penetrance, the penetrance is what? It is the percentage, it is the percentage of those organisms in a population, is a percentage of those organisms in a population which shows phenotype associated with their genotype. So it is a percentage of organism that shows phenotype associated with genotype. Means genotype is present in 100 individual, but it is getting expressed or it is forming a protein or it is showing a trait in only 80 individuals. Then the penetrance is just 80%. Okay. So the penetrance is of two type. Number one, complete penetrance. Complete penetrance. Complete penetrance means 100% individual show the phenotype associated with the genotype. Whereas another is incomplete penetrance. And in this incomplete penetrance, if you talk, it is less than 100%, means less than 100% of individual show phenotype associated with the Genotype. Okay. So if suppose 80% are showing the phenotype associated with the genotype, then that is 80% penetrance is found. Okay, so this is the thing for this particular penetrance. Okay, so this many terminology we have discussed. Now we are going to discuss some more thing regarding this. Okay, so let's talk about some of the characters which are controlled by a single gene, or a traits which are controlled by a single gene. So single gene trait is one. The example are clasping hand, tongue rolling, hitchhiker's thumb, earlobe, bent little finger, etc. And we are showing this all by diagrammatic pattern. See, clasping hand means the joining hand. Okay, then free or attached earlobe. Okay, see, first one is the attached, second with the free. Then hitchhiker thumb means thumb is somewhat bended like this. Okay, then rolling tongue get rolled by like this. Okay, then dimples in an individual. You can remember Shah Rukh Khan. Okay, then father and his daughter with identical facial features. See, father and daughter is showing the same features, identical features. Father and son taken photograph at the same age, they show a similar characteristic. Father and son, father in 1937 and the son in 1965 taken a photograph at same age, they show some similar features. Identical twins, that is also due to the genetics. Okay, identical triplets are also due to the genetics, and identical quadruplets are also due to the genetics. So these are the examples and for which the genetic is responsible. Now, till now, whatever the terminology is going to be used, what is genetic, what is in variation, the introductory part we have covered. Okay. Now onwards, actually, we will going to discuss the genetics. And in that, we will be talking about pre-Mendelian, Mendelian, and post-Mendelian genetics in the next lecture. Okay. So before studying Mendelian experiment, revise this whole thing. Then we will start with the Mendelian experiment and that process. Okay. Till that, thank you.